Dr. Possu Huang received his PhD from Caltech with the first demonstration of computationally designed novel protein-protein interface. He conducted postdoctoral research at the University of Washington before starting his group at Stanford. His research focuses on advancing the understanding of proteins for the engineering of novel therapeutics and other protein-based nanotechnology. Tell me if you need me to stall more. He has contributed to a large number of de novo designed proteins, most notably to the unlocking of the design principles behind the TIM barrel fold and the invention of EOD and HIV immunogen design. His group uses machine learning, computational modeling, structural biology, and experimental library optimization to continue the expansion of protein-based molecular platforms. And I turn it over to you, Possu.
Thank you for the introduction, and thank you all for hanging out until the very end. So the first thing I would like to say is that thanks for the invitation, and then you can definitely tell that the next talk is an academic talk, because my picture is not as good looking as Gevorg's nor Raphael's. So, but I will try to see if we can actually bring something fresh to your mind in terms of this area. So I'm going to talk about this topic on the latest research that we do on proteins, and people are probably wondering like, what's this word like PROTPARDELLE, but hopefully it will be revealed at the end. So in the last 10 years, this is just a picture that I took from the review that we wrote a few years back, and now as Gevorg has mentioned, that we have the ability to build structures from scratch, essentially. And then these pictures are actually back in the days, we call it like, it feels like it's Stone Ages, because we're essentially building everything by hand. And nowadays you can actually use machine learning to actually take a lot of the jobs automatically. But today's talk, I'm actually gonna focus a little bit. Actually, we didn't pre-arrange this topic, but Gevorg's talk essentially dovetail into what I'm trying to describe today. So the goal here is that now we have the ability to handle these structural aspects of proteins, but how do you really get at functionality? So my lab has been spending the last few years trying to get at this notion of using protein dynamic structures and to get at function, and at the same time, we're also trying to perhaps build features that actually allow us to break like the current boundary of the tools that we have today. So we proposed this way of thinking about this problem as a new central paradigm for de novo protein design. So essentially, this is pretty intuitive, right? Like you started out with a protein structure, with a function that you want to achieve. And then again, Gevorg introduced this very, very nicely, that you normally would have to build a structure to hold it. And then also this a third track that actually has a sequence component. You would have to decorate these structure with sequences, and then lo and behold, at the end, hopefully you will have a protein that you can actually test in the wet lab. So I'll actually give you a few examples on how we could potentially achieve function, and some of the efforts that my lab has introduced on the sequence side, and then also the structure side. So and then I'll introduce this PROTPARDELLE business. So a few years ago, actually it's only 2022, I feel like it was like a few years ago already, but so we actually try to design a scheme where you can actually introduce side chains of proteins so that you can actually directly design for function. So this choice, we essentially take a side chain, a protein, and we build a molecular representation that is just simple point clouds. And we use it to predict the probability of residues for a given chemical environment using this point cloud representation. And when we first tried this, it was suggesting that by learning this type of classifier, you could actually essentially mimic energetics of a protein design energy functions. So I'm not going to go into details here, but this representation is chose very intentionally, because now with this point cloud representation, you could easily adapt to any ligand, essentially. And the example I'm going to show here is that we use this way to design sequences and try to incorporate metal binding sites. So it will be a very convenient scheme if you actually can represent metal coordinates, like by describing the property and hybridization of these metals and oxidation states. You could literally just assign in your point cloud representation occupancies of the area where the metal sits, right? So when you mix these two components together, now we actually have this fully automated metal binding design algorithm, where you can kind of see that you could just have a starting protein scaffold, and it can directly design metal binding site and can refine the positions. Because the previous picture I showed you was actually a metal site predicting algorithm. So we now have a sidechain design iterating over this metal position predicting algorithm. You can kind of see that you can scan for positions where all the geometry can hold the metal, and we can design this way. So the reason I went into this detail to try to portray this ability to design function is actually related to this question about inference versus generalizability of these protein models. So generally, nowadays, we have seen a lot of evidence of a very powerful model that can actually do inference. You've probably seen like RF diffusion that can directly diffuse a helix that sits on top of some binding site. You can make a protein to host it. But it turns out that a lot of these inference, and actually I think from an earlier question for Gavor, was getting at this notion, how do you actually generalize to function if you have instances that are not actually been observed before? So the way my lab look at this problem is that we actually strive to build all the models that we build are try to generalizable to any problem. So this is actually one interesting example. This is a slide from my friend Bruno Correa from EPFL. So they were trying to design GPCR folds. These are not functional GPCRs, but they just try to solubilize it. But then they were testing the various sequence design algorithms and see if the success rate for each of those. Okay, so it turns out that with the conventional methods, Rosetta, and some of the machine learning methods, it could not actually predict a very good sequence that will actually fold this type of structure and be soluble in solution. So this LP is actually the method that came from my lab. We directly use the voxelized representation. So you actually learn the chemical environment. And it turns out that right out of the box, you could actually create this type of structure that's soluble. And these other two bars are actually really, really interesting because recently David Baker's lab came out with this protein MPNN algorithm. And essentially, if you directly use a protein MPNN algorithm, it memorizes the GPCR that you have seen before. So it actually gives you hydrophobic surfaces, even though you present the protein as a soluble protein standalone without the membrane. But if you fine-tune MPNN on some helical structures to basically try to erase this memory of GPCR being a membrane protein, then your success rate goes way up. So this, again, comes back to this question, how do you actually build a protein that actually would be generalizable to these different type of problems? So maybe perhaps take a step back to this question of the central dogma on protein design. And then perhaps before I get into like how to deal with this function for a very generalizable case, I will go into another example on the structural side of design. So early on, again, 2018 really feels like a century ago. My lab was like the first lab that puts out a generative model on protein structure. And back then, we'd have no idea what we were doing. We literally just borrow all the algorithms from the imaging side. And we essentially train again and then make a fake protein 2D dimensional distance matrix. And then we can recover the structure. And then we think that this could potentially allow us to build new de novo protein structures. And the movie in the middle right here, of course, it doesn't look like any real protein. It looks terrible, and you would absolutely not ever want to make any effort to try to produce in the wet lab. But this movie actually shows us one very, very interesting aspect. And actually, that's also the point that Gavor has made a point on. A protein design problem is essentially a constraint satisfaction problem. So in the middle, the movie in the middle essentially is that we injected hydrogen bonding network calculations into the model, and we actually use the latent space of GAN to optimize the structure. And you can kind of see as you search the space, you sort of improve the structure to something that looks somewhat folded. Okay, so those are the early days. These proteins are just looking terrible, and of course Gavor's cases are much more impressive. But my lab also has focused on developing methods to develop this general functional design of proteins. And the one question that we want to answer was to see if we can actually contribute to the area of antibody design. So if you really just list the specs of what you have to satisfy, in order to build a protein-protein interface. The driving force is generally the shape complementarity along with chemical complementarity. So we come up with this idea, perhaps we can leverage generative models to actually shape the protein so that we can actually mold them to the target. So the concept here is that if you have a complex topology, already, if you want to satisfy this shape complementarity and chemical complementarity, perhaps using a generative model will allow us to actually very quickly make proteins that actually have the complementary shape. And then coming back to this notion of a constraint satisfaction problem for a protein design, early on we using this model called variational autoencoder, and we directly generate these coordinates from the model, and it does allow us to actually constrain to any environmental factor that we can put in. So in here, it's just an example of showing a loop. You could actually constrain on the loop, and then the generative model can actually bring up a IG full to actually host it. But this is actually the easy part, right? Because building a generative model, we have a lot of experience. It's actually quite easy. But the second part is where I was getting at, where we need to come up with a new solution so that we can actually address this issue, how to form interface between two molecules, right? So this is actually the way to actually create contacts on the surface so that you can create a binder that can satisfy these constraints that you put on is actually much, much harder than any of these ML tasks. And also my lab essentially think at the time that we wouldn't have enough data to train this type of model. So we actually leverage a balance between biophysics and machine learning. So the scheme that we came out with is this algorithm called Sculptor. So what we do is that you can essentially take a target of interest, you can assign wherever you want to bind to it, and we can actually leverage data to create proposals to actually make the contacts to the surface. And all we had to do for the generative model is essentially just to, kind of like the movie suggests, to sculpt a protein by optimizing it against these proposal residues. So what you come up to be is essentially you have a structural sampling algorithm. So it's a fully conformational sampling algorithm with a flexible backbone. And at the same time, you have some gradient guidance in order to actually optimize, to approach the target. So this, with this strategy, we actually could build binders to practically anything, or at least on the computer, we can model anything. You could actually have the same strategy to bind protein to protein, and you can actually use protein to bind to RNA. And actually, if you have a generative model that's RNA, you can also just flip it to use RNA to bind anything. So experimentally, we validated this to some degree. We definitely have to do more. And this is just an east display, just to guide your eyes. So if you have any population that's on this upper quadrant, that means we have positive clones. So these are actually libraries of explicitly designed sequences. So it's not a directly evolution experiment. And for any of these targets, we could actually get binders directly coming from the computer. So the implication of this, and this is what we are currently working on right now, we're extending from this one simple domain to larger solutions, for example, a monoclonal antibody. So this is a summary taken from a review recently, and these are the current methods of creating monoclonal antibodies in biotech. And we're now hoping that perhaps we can use this strategy to directly make antibodies in silico. And of course, the people will say, if you can easily make, because now antibody pipelines are very, very mature, what is the point of creating these things in silico? So first of all, I think to hit a very specific epitope is difficult. And also, this could potentially open up possibility that you can actually create conditional activating antibodies. So this one, just for fun, we actually take our antibody sculpting trajectories and put them together. It always reminds me of something like the piranhas on a speeding frenzy. So anyway, so I'm going to run out of time, but hopefully I can get to this all-atom model that was part of the title. So if we come back thinking about function, and if you look at this central dogma of arranging these hierarchical structure of proteins, perhaps it's actually not necessary, because the reason why we set it up this way is actually a conceptual barrier where you really treat side chains independently from a backbone. But we're actually trying to address this problem perhaps where we could actually come up with a joint model where the sequence and structure can be jointly optimized. So if I use a visual guide to illustrate this point, essentially if you think about, say, coordinates are actually these diffusion models that Gevorg has introduced, essentially coming from the upper right-hand corner, that means you don't know anything about structure and you don't know anything about the sequence. You want to get to the bottom left of everything that's denoised. So you could go along the structural route or you can go along the sequence route. And all four corners actually have respective algorithms that already address them. So for example, this, well, I would say perhaps this track is actually the easiest one. So you have, so it's at the very bottom of the sequence, so you already know the sequence, but you want to try to get the structure of a given sequence, so that's basically AlphaFold. Okay, so, and I don't have to go into this because Gevorg already gave you the background on the diffusion, but what's really powerful about this diffusion model is that it allows you to actually capture the information flow in a complex system like the sequence structure dimensions. So we come up with a arrangement. Essentially, I'll just skip forward. So we essentially try to use a diffusion model to actually capture this information exchange. And the reason, and actually one of the first structure that came out of this model actually looked like this. And that actually is the reason why we call this algorithm PROTPARDELLE, because it's essentially the pasta PROTPARDELLE. So now you know why it's called PROTPARDELLE. But what's unique about this algorithm is that it's actually a language model. So we're actually using language model to capture every atom and describe atoms into residues, and then using residues to connect them into a polymer chain. And because we're actually capturing this atom level modeling, we actually can introduce this new concept that directly builds sidechains. So as Gevorg mentioned earlier, that usually the current state of the art generally builds backbone and treats sidechains separately, even though they're crosstalks, but there's no explicit model that builds you sidechain directly while you build a backbone. So it came up with this concept called superposition. Essentially, every residue we're diffusing carries all the atoms that can morph into any structure and any conformation. So as you're denoising, you could actually basically resolve masks so that you can actually collapse these residues into a different conformation of the same residue, or you can actually make mutations. So this is just a sort of obligatory diffusion picture, and the only reason to show this is so that you can appreciate that this is actually an atom-based model where we're actually driving it without actually constraining on residue or any chemical connections. And I'll skip this very quickly. Essentially, we can build de novo structures, just like what Gevorg showed, and then these are just showing that if you look up the nearest neighbor in the database, they're actually not, there's no nearest neighbor in the database, even though their parts are similar. So in the last two slides with this punchline is that the reason why we get at this problem, why do we insist on putting sidechains into this type of models, and also want to have this co-design capability is actually this bottom picture here. So what we really want to achieve is that if we have a model that's powerful enough, you could potentially just have the functional groups of a metal binding site, and we purely constrain on the geometry, and the model should be able to actually get you a protein that can host this entire thing. So, and then also, of course, this type of model is very flexible, exactly like what Gevorg has introduced, and nowadays we're actually using it to design something that actually very few instances exist in nature. It's the cyclic peptides. So we could actually use this model in conjunction with a sculptor algorithm to actually make this type of molecule. And here we actually modify it a little bit. So it's no longer the sculpting using a VAE, but it's actually every frame when the peptide is jumping around is actually a diffusion step. So we could actually run this incredibly quickly. So in summary, no, okay, I'm just jump to acknowledgements. So one other thing you can also tell that this is an academic talk is that Gevorg has like a lot of really talented engineers, and I have like 3 graduate students. But anyway, but my students are very talented, and they actually were able to pull this off, and has these really interesting features that we're now just at the beginning of addressing. So and thank you for your attention. I'm happy to take questions.
let's keep it to maybe 2 questions maximum.
Thank you. Great talk. So for integrating sequence to structure-based model, how about we use the hidden Markov chain to extract information as an additive latent space layer?
Yeah, we could do that actually. So, but usually this type of model, when you train, there are actually folded structures. We could do it if you actually pre-train a data on like some sort of random chain. But generally we don't do that just because it's too hard to manage. It's not tractable essentially. Yeah. It's kind of the similar question about the intrinsically disordered protein that was directed at good work. Yeah, they are actually difficult to do. Yeah.
Thank you.
Yeah, I guess to the intrinsically disordered protein question. I mean, I think for even just for a very structured protein, it makes sense that you could fit a binder conformationally to that. But even in the in a cell or in any environment, there will be unique conformations that protein can adopt, and some proteins will be more disordered. What do you think about what would you expect if you were to use the epitope-specific binding strategy you described for proteins that are maybe not fully disordered, but at least a little more disordered?
No, I'm pretty sure we could do it. So if you have a peptide generative model, we could totally dock them and design them the way I described. Because like I say, it's actually very flexible. You can use any generative model and hook it up to that scheme. And actually, the reason why we start looking into these cyclic peptides is kind of like a compromise between flexible structure and the protein. So this type of model, we hope that we could actually make really effective cyclic peptide drugs. Yeah.
All right, thank you very much, Possu.
Thank you.